Well, we're talking about membranes. Of course, we just spent a lot of time looking at their structure and what the properties of the lipid bilayer are. And of course, then we talked about the fact that 50% of the membrane is actually made up of proteins. And we saw how the proteins can be associated with the membranes, particularly association through linkage to a fatty acid, but more importantly, many proteins are transmembrane proteins, which have these transmembrane domains, which hold them into the protein through the lipid bilayer, since the transmembrane domain is a nonpolar alpha helix. But anyhow, I want to start moving on and talking about the types of proteins and what some of the things that they do. And of course, they do a lot of things. There's receptors, and there's pumps, and there's channels, and we'll talk about those. But I want to start out talking about certain types of membrane proteins that are called cell adhesion molecules or cell junctions. And these are proteins that allow cells to attach together in order to make a tissue, or they attach to some substrate, like some kind of a carbohydrate substrate, in order to build the basis of a tissue. And so we're going to talk about really three different classes, and this will give you a pretty good idea of what they look like and how they function. So there's tight junctions, and the tight junctions are responsible for not just holding cells together, but sealing cells together so that nothing can pass between them. And then there are anchoring junctions, which are used to hold cells together. Also, sometimes they're used to hold cells to some kind of a surface, but they're not serving the same function as the tight junction. They can allow materials to pass between the cells. It's really just to hold them together. And one of the properties of the anchoring junctions is that they have what's called a cytoskeletal support that is on the interior of the cell, on the cytoplasm side. They're linked up to the cytoskeleton to give them more support. And we'll look at some of those structures. And then finally, the gap junctions, they're not really there to hold the cells together, but they do participate in holding cells together. But more importantly, these junctions allow the transfer of molecules between cells in a tissue. So let's start looking at these. We'll look at the tight junctions first. So here we're looking at tight junctions, and they make a really leak-proof seal between cells, and here they're showing epithelial cells. But you'll see it also in cells of the gut, where you don't have material flowing from the stomach out into the abdomen. The cells are so tightly held together that nothing can pass through, no liquid could pass between them. So they're made by proteins called occludin and claudin, two different types of proteins that interact with each other. And if you look at the drawing on the right-hand side, what it's showing is kind of like a cross-section where you have two cells, and we're looking at the edge of the cell, and you can see the green little dots represent the adhesion of these two proteins together. Now, if you look carefully at that diagram, what you see is running along where those green dots are, there's a whole series of little circles, and what those represent are the clustering of these occludin and claudin proteins, and making this, and they're so close together, and they bring the membrane so close together in forming this junction, it almost looks like stitching. And so it holds the cells together, and it makes this incredibly tight and impermeable junction. And now on the left-hand side, there's a few electron micrographs that show this. The one that actually really shows it, I think, pretty well is the one on the right. shows cell 1 and cell 2, and then that tight junction is that dark strip which is sealing those cells together. Nothing can really penetrate between those two cells once it hits that tight junction. So those are tight junctions, basically to adhere cells together so tightly that nothing can pass through them. So I want to talk a little bit more, or a little bit, about what are called anchoring junctions. And that's shown in the next slide. So here we're looking at a type of anchoring junction which is called an adherence junction, and the adherence junctions are, the purpose of those is to hold cells together in a tissue. It allows some movement, some flexibility, but they hold cells together. They're not held as tightly as the tight junctions, but they hold, their purpose is to hold cells together. Now, so let's take a look at this figure. Looks like there's two or three cells that we're looking at, and I believe the type of cell that we're looking at is a cell from either large intestine or small intestine, because you have these microvilli up at the top, and microvilli, pass the food around, or the solution, and then you have the lumen, which would be the interior of the intestine or the interior, of the colon. And then you have at the very bottom, you have the basal, what's called basal surface. So these cells are held down to a matrix that holds the cells in place. Now what you can see are the membranes. It says lateral plasma membranes of adjacent epithelial cells. And then as you move up, if you look specifically in the middle, you see the adherence junctions, and if you kind of can enlarge that, you can see that they're held together by proteins which are called cadherins. And the cadherins are attached to bundles of actin filaments on the inside and the cytoplasmic side, which lends support to these things. And we're going to look in a minute at another type of anchoring junction, which is called a desmosome. The principle is kind of the same, but the drawings illustrate it a lot better. So here we're looking at some really nice images of desmosomes. And desmosomes are, in a sense, analogous to the adherence junctions. The adherence junctions are bound on the interior of the cell by actin filaments. The differences, and one of the differences between the adherence junctions and the desmosomes, are that the desmosomes are supported by keratin filaments. So it's still cytoskeletal proteins which are backing up or supporting these structures. So let's look at the electron micrograph first, and you can see cell 1 and cell 2. That dark region, it's not like the tight junction because you can see the space actually in between them. And the proteins which are holding these cells together are called cadherins. And the cadherin proteins are the same proteins, the same family of proteins that hold the adherence junctions together. The cadherins are a large family of transmembrane proteins that are specifically involved in holding cells together. There's lots of different types, but basically they're quite similar. So they're found in many different types of junctions. So if we look at the right-hand side, this is actually, I find it a pretty good drawing. It pretty much explains what's here. So you have the two cells, cell 1 on the left-hand side, cell 2 on the right-hand side. We're looking at the plasma membrane of those cells. You can see they're held together by these cadherin proteins. The cadherin proteins are single-pass transmembrane domain proteins. and they interact. I'll show you a drawing a little bit later. They interact through their amino terminus. They self-assemble. Just like we talked about the quaternary structure where things self-assemble, these things have complementary surfaces that allow them to attach to each other. So within each desmosome, you may have a couple hundred of these cadherins which interact with each other, and then they make that junction. Now they pass through the cell membrane with a single transmembrane domain, and so then on the inside of the cell, on the cytoplasmic side of the cell, there is the carboxy terminus of the protein, and they are held and supported by what are called catenins. Now, and then the catenins are attached to keratin, and that's what's represented by the blue. So the keratin filaments are anchored. They're calling it a cytoplasmic plaque. You can call it that, but the proteins that make up the cytoplasmic plaque are called catenins. And so what's happening here is the cytoskeleton, which in this case is keratin, are supporting the structures that are holding these cells together. So the way I think about it is if these cadherin proteins were not backed up by any cytoskeletal proteins, it would be very easy for the cells to be pulled apart. Because you pull them apart, you put a little bit extra tensile strength on that, and then the cadherin proteins would come right out of the membrane because it's just a single transmembrane domain. They just pull right out. So what's happening here is they're being supported by the cytoskeleton, so you can't pull them out. They're anchored in very tightly, and they're held through the catenins to the cytoskeletal keratin proteins. So the next figure shows a little bit better, and it also has one of my drawings. So here, this kind of shows it in a little bit more detail. It shows the cadherin proteins. Look on the left-hand side. It shows the cadherin proteins, single transmembrane domain protein. Of course, in a desmosome or hemidesmosome, you'd have hundreds of these going through, but they're just showing two. One coming out of cell 1, one coming out of cell 2. They interact through their amino terminus. And then on the interior of the cell, on the cytoplasmic portion of the cell, the carboxy terminus interacts with the catenin. That's what I showed you before. And then that catenin is then interacting or bound to the keratin filaments. So three proteins here. You have cadherin, you have the catenins, and then you have the cytoskeletal proteins. And so I'm showing that again on the right-hand side in my drawing, which is basically kind of the same thing, showing three different cadherins interacting through the amino terminus, single transmembrane domains. In the top two, you can see that if you were to pull the cells apart, those things would just come right out of the cell membrane. But on that lowest one, where I have the carboxy terminus interacting with the catenins, and then, of course, interacting with the cytoskeleton, that provides much more support for these cadherin proteins in order to make these junctions. Now, I want to go off a little bit on a tangent. Actually, let me back up for a second, just to say one more thing about the catenins. What happens here is that the catenins attach to the carboxy terminus of the cadherins, which are these proteins that hold cells together, but the catenins also interact with each other. So what happens is when you get a cadherin bound to a few catenins at the carboxy terminus, and you have this happening at a few different places along the membrane, then these catenins actually will interact with each other on the different cadherins and allow them to cluster into these desmosomes. So the catenins actually bring the other ones together, and it causes them to cluster into this nodule that then holds the cells together. But I want to digress a little, not digress, but just kind of go off on a little bit of a separate tangent for just a second and talk a little bit more about catenins, because the catenins turn out to be multifunctional proteins, and what nobody ever expected is that they play a very, very important role in colon cancer. So let me tell you about that. Okay, so what I've written here is beta-catenin. It's a multifunctional protein with a role in colon cancer. Let me explain what happens here. So this represents a cell membrane, just kind of a square. And what I'm showing here is like an adhesions junction on the right-hand side. So you have the cadherin, which is passing through the cell membrane. Of course, in adherent junctions, you'd have lots of those clustered together, but I'm just showing the one. And then on the carboxy terminus, it's attached to the beta-catenin. And then, of course, to the actin cytoskeletal proteins, which acts as that support. Now, the beta-catenins don't last forever. They get replaced. So there's a constant turnover. And the way they get turned over, this is basically what I told you when we talked about the post-translational modification and the degradation through the proteasome. What happens is the beta-catenin gets fed into the proteasome. It first interacts with a protein called APC. APC is a ubiquitin ligase, just like we talked about the ubiquitin ligase P53 and some of the other proteins that get targeted to the proteasome. So what happens here is during the turnover of the beta-catenin, it gets attached to the APC. APC is the ubiquitin ligase. It adds the polyubiquitin to the beta-catenin, and then that targets it for the proteasome. It goes into the proteasome and gets degraded. Now, under some conditions, in response to certain growth factors, the beta-catenin does not interact with APC, and it is modified and sent into the nucleus. That's what I'm showing at the bottom. So it happens only in response to certain growth factors. So rather than getting replaced and ubiquitinated, sent to the proteasome, it gets sent into the nucleus, and it becomes a DNA binding transcription factor, which nobody ever expected. Binds to DNA, and particularly it binds to the DNA, the promoters of genes that activate cell division. So it turns on genes that activate cell division. So here we have an example of a multifunctional protein. It acts as part of cell-cell junctions, interacting or causing the interaction and the binding of the cytoskeletal to the cadherin proteins. And the truth is, nobody ever thought to ever look for anything else. I mean, it was involved in cell-cell interactions. Okay, we got it, done, move on. Nobody was studying beta-catenins anymore. It became kind of like an old story and a little bit boring, and okay, we got it. And then people looking at cell division, all of a sudden found that some genes were activated by beta-catenin. It's like, that's impossible. No, it's not impossible. That's what happened. Multifunctional proteins. I'll talk more about them in a minute. So that was, first of all, an eye-opening finding that a quote-unquote boring cell-cell adhesion protein is now activating gene expression and cell division. And then it got even more interesting when it turned out that the APC gene, that's the ubiquitin ligase for the beta-catenin, the APC gene is mutated in 80% of all colon cancers. So 80% of all colon cancers carry mutations in APC. The mutations in APC inactivate the ubiquitin ligase, so it's non-functional. So the beta-catenins accumulate in the cell, and then some of them get into the nucleus and then activate the genes that are responsible for cell division and cause the cells to divide. So mutations in APC lead to colon cancer because they allow the beta-catenin to inappropriately activate these genes. So that's basically what's shown here. Defective APC leads to high levels of beta-catenin and the deregulated gene expression, cell division. So here you're showing the APC is not working, the beta-catenin accumulates in the nucleus, in the cytoplasm, it's overexpressed, gets into the nucleus, activates gene expression, cell division. So colon cancer is basically due to the overexpression of beta-catenin, which people for many years didn't pay attention to because it was just a run-of-the-mill cytoskeletal type protein involved in cell-cell interactions. And then all of a sudden we start seeing other things that beta-catenin is involved in. Let me show you this one here. So this was published a couple of years ago, and if you look down towards the bottom there, I kind of marked it off on the edges. This came from, I think this was from the journal Nature, and it's talking about the role of beta-catenin in depression. So that's expression in the brain causing some deregulated neurotransmitters that is then leading to some types of depression. So going one more tangent. So the first tangent was basically we're talking about cell-cell interactions and proteins that are involved in cell-cell interactions like the cadherins and the role of beta-catenin in stabilizing those cadherins. And so the first tangent was to say, well, beta-catenin actually has multiple roles, multiple functions in the cell. It can also be a transcription factor. And then another little tangent was that, when you have mutations in APC and overexpression leads to colon cancer. So it turns out that proteins often have multiple functions and many that we're not aware of. And in terms of the psychology of science and the way experiments are done, names can have very significant impact on how we think about functions and the possibilities for other functions. So you call a protein, DNA polymerase. And people have a very hard time imagining that protein doing something else because it's called DNA polymerase. Or you have a protein called catenin, which for years people knew what it did, and all of a sudden, it's doing something else. So the name of the protein, or the name that's given, often is an impediment to looking at other possible functions. It's like we shoot ourselves in the foot, in a sense, by naming these things, because the name implies that that's the function, and then that's the only function. But I'm going to give you some examples of other proteins that have distinct multiple functions. It prevents research from being done in a sense. So here's examples of proteins with multiple distinct functions. So beta-catenin, of course, we know. We've talked about a cytoskeletal protein, cell adhesion. It's in the cytoplasm, and recently found that it can regulate transcription in the nucleus. Here's some other examples. Cytochrome C. Cytochrome C is part and parcel of electron transport in the mitochondria. That's what it does, and that's what it's always done, and that, nobody was interested in really studying it anymore. So we figured, okay, we know what it does. And then all of a sudden, people who were starting to study apoptosis, programmed cell death, found that cytochrome C, the same cytochrome C that is electron transport, the same cytochrome C gets out of the mitochondria and then participates in activating a series of proteases that precipitate apoptosis. So it has multiple functions. Here's another one, which is even more, not even more striking, but you can see how people can get stuck. So this is ribosomal protein L41. Involved, of course, in protein synthesis and the ribosome and the cytoplasm. Nobody ever in their right mind thought that the ribosomal protein L1, which is a part of the ribosome, can also get into the nucleus and regulate transcription. See, that's the problem of the names. You have ribosomal protein L41, done. Nobody wants to study it. You try and study it, you can't get grant funding. You come up with some hypothesis, nobody's going to believe you because they're stuck on ribosomal protein L41. So you got to go the extra mile and show that this same protein is involved in regulating transcription in the nucleus. STAT3 is another one, transcriptional regulation in the nucleus, been studied for years. Turns out it's involved in the mitochondrial electron transport. Then crystalline is another amazing one. Crystalline is involved in light transmission in the eye. Crystalline is a protein that when it aggregates, self-assembles, it aggregates, it makes the lens of our eye. It's clear. The clear lens is made of crystalline protein. So it allows light to go right through it. It's made from protein. But nobody ever would have expected that it's the same exact protein as lactate dehydrogenase, which is involved in glycolysis in all cells. So people studying lactate dehydrogenase were not interested. It's like, okay, we know what it is. That's the end of that. And then it turns out it's the same protein that we use to see through our lens. So it's just an example of how, it's just psychology. We name proteins for what they do, and then it prevents us quite often from studying these proteins, many of which may have multiple very important functions. So I just want to close here and talk about another type of cell-cell interactions, and these are called gap junctions. Gap junctions, they do hold cells together, but their role is to allow the passage of molecules between cells. So here we're looking at gap junctions. If we look on the left-hand side of the electron micrograph, they're saying they see cell 1 and cell 2. They're calling those gap junctions. I mean, I can't tell from looking at that. Looks like any of the other junctions that we've looked at. We get a little bit more information on how these things are structured by looking at the right-hand side of the drawing there. So the gap junctions, what happens with these gap junctions is they allow the passage of molecules and signals between cells very rapidly. They're made up of connexin proteins. These are transmembrane proteins, connexin proteins. They're made they're made up, each cell has what's called a connexon, made up of 6 of these connexin proteins. So we're here looking at 2 cells, one on top we call cell 1, one on the bottom called cell 2. We're looking at a very small area of the plasma membranes on both of those cells. So the connexin proteins cluster together to form a connexon on the 2 different surfaces of the cell, and then they align themselves up and they link up to form. And the other thing I should mention is that connexins have a passageway, a pore, that will allow the passage of materials. And so when the connexins of 2 adjoining cells link up, you get a series of what are called these gap junctions, where every one of these has a passage that allows the passage of small molecules and signals, hormones, ions, which participate in signaling one cell to the next. So gap junctions are found particularly in cells that need to communicate with each other rapidly. It's almost as if the cell, if you have a cluster of cells with many gap junctions, it's almost as if it becomes one cell, because the information that's passed to one cell is then passed to another cell through the gap junctions extremely rapidly. And so we often see these kinds of gap junctions in cells that function uniformly like muscle contraction, particularly in the heart. We also see it in the uterus a lot. So you can imagine in the heart, where there's lots of these gap junctions, there's a particular signal that hits one node of the heart, and it doesn't need to, that signal does not need to hit the whole heart. It just hits one node of the heart, and then the cells respond by contracting, but the signal to contract is passed into all the cells very quickly, so you can get this pulsating contracting of the heart muscle. Same is true with the uterus, and that is there's lots of gap junctions, so you can get the signal at one portion of the uterus, and that signal is rapidly transferred to all of the cells of the uterus, and it allows the uterus to contract, which is what happens during labor when a woman is giving birth. So bottom line is they're required for rapid transfer of material and information between cells. So that's going to be it for today. So today we saw 2 presentations. One, we started out looking at membranes, membrane structure. Then we moved into membrane proteins, at least how the membrane proteins get there, what they look like, how they're held in the membrane. And now we're starting to look at different functions of different membrane proteins. And today we looked at particularly at those proteins which are involved in cell-cell interactions and holding cells together, the cadherins, and then also the connexin proteins, which make up the gap junctions. Okay, so have a good rest of your day.